Hello, everyone. Good morning, good afternoon, or good evening, wherever you're joining us from. I'm Khachaturian, and on behalf of the Campaign to Prevent Alzheimer's Disease, the Brainwatch Coalition, and the International Neurodegenerative Disorders Research Center, welcome to episode 4 of our series, Empowering Proteomics and Transcriptomics in AD Drug Discovery. Today's episode is titled Unraveling Alzheimer's Heterogeneity, Clear AD, on the road to plasma biomarkers.
And I'm Ornit Chiba-Falek from the Division of Translational Brain Sciences in the Department of Neurology at Duke University. As always, we want to thank Frontier in Molecular Mechanism of Neurodegeneration for their generous support in helping launch and sustain this series. In this series, we have been pushing the conversation beyond the current state of Alzheimer drug discovery and biomarker development towards a biological mechanism and translational hurdles that will shape the next decade of Alzheimer and dementia trials.
In our second episode, Paul Azen helped us reflect on what we've learned from landmark trials like A4 and AHEAD. In our last episode, Russell Swedlow guided us through biological mechanisms and mitochondrial cascade hypothesis and discussed with us how omics are the compass in navigating the road from biological mechanisms to personalized medicine. Today, in episode 4, we're asking how do proteomics and transcriptomics not only generate discovery, but also amplify efficiency and accuracy in dementia drug development? That question was at the heart of our October 2024 editorial in the Journal of Prevention of Alzheimer's Disease, and it's the springboard for our conversation today.
Yes, this is the inspiration of today's conversation, and we are especially excited to welcome our guest, Dr. Nilüfer Ertekin-Taner, because she has spent her entire career at that intersection, connecting between multiomics discovery and patient care through defined molecular endophenotype. Dr. Nilüfer Ertekin-Taner, a longtime colleague, is the Roy E. and Merle Mayer Professor of Neuroscience and a Professor of Neurology at Mayo Clinic Florida. She also served as an enterprise chair of neuroscience and a director of the RNA therapeutics program at Mayo Clinic. In addition, she is a practicing behavioral neurologist and internationally recognized neuroscientist. As a physician scientist, she will bring into the conversation the bench-to-bedside perspective for complex and heterogeneous diseases such as Alzheimer and Alzheimer-related diseases.
Exactly. Her pioneering work on endophenotypes in Alzheimer's genetics using amyloid beta peptide levels to uncover risk pathways has shaped how we think about translating molecular signatures into clinical research. She leads major NIH and multi-site collaborations, including AMP-AD, MOVE-AD, Resilience AD, the Florida Consortia for African American Alzheimer's Disease Studies, and most recently, CLEAR AD. Dr. Ertekin-Taner, welcome to the podcast.
Thank you. Thanks for having me.
Nilüfer, it's a real honor to have you with us. You have helped defining some of the most important genetic determinants and molecular and biological pathways in Alzheimer's through basic science research. You have also one foot clinic. That dual perspective is exactly what we would like to highlight today.
Exactly. At this stage, let's start by defining what we mean by molecular endophenotype in this conversation, because that's where efficiency and trial design may begin. But before we dive into that, into what's next in Alzheimer's biomarker development, let's pause on where we are today. This year, the FDA cleared the first blood-based test to help assess amyloid pathology. This is the Lumipulse GP tau 217 A beta 1 to 42 plasma ratio for adults with cognitive impairment. It's less invasive and expands access, but it still infers plaque pathology and usually requires confirmatory testing. Nilüfer, from your perspective, what are the main benefits and drawbacks for this test for clinicians and patients?
Sure. Thank you for the question, and thanks for having me, Ara and Ornit. It's a great pleasure to be having this conversation with both of you today. So in terms of biofluid biomarkers of Alzheimer's disease, we have come a long way. Indeed, you referred to some of our earlier studies where we have pioneered the use of plasma amyloid beta as an endophenotype in genetic studies of Alzheimer's disease. Those early studies taught us that whether we're talking about early onset familial forms of Alzheimer's disease or the so-called sporadic late onset forms of Alzheimer's disease, plasma amyloid beta may be a very useful endophenotype for genetic discoveries. Those type of studies from our group and then subsequently by other groups have paved the way for the biofluid biomarkers that we are using today, where which culminated in this FDA approved plasma biomarker. So Ara, I think what you're referring to is, are we at a crossroads where we have this capability of being able to determine with some certainty whether somebody has amyloid in their brain or not, and how do we use it? What are the benefits and what are the drawbacks? I think we need to be very careful about the context of use under which FDA has approved the use of this biomarker, as is the case with any biomarker. I think you started your statement by saying that for adults with cognitive impairment, but having a blood test so accessible at our fingertips, it may make it easier to simply use this test beyond the context of use. But after evaluating these patients, they do not have evidence of cognitive decline. So the question is, is that applicable in this scenario, which it isn't. I think we also need to think about the actionable outcome from the test. So some actionable outcomes might be you have a patient, they have cognitive concerns, you put them through objective testing, and let's say they have mild cognitive impairment. They have cognitive impairment, objectively established, it's not yet interfering with their day-to-day lives. And knowing that this mild cognitive impairment is due to an Alzheimer's type pathology in the brain, meaning presence of amyloid plaques, that could be an actionable piece of information. How is that actionable? Well, if that patient is willing, they might be eligible for the newly approved anti-amyloid therapies, provided they hear the risks and benefits. So that's a scenario where you may want to proceed with a screening blood test first, and then follow that with the more confirmatory testing, either through cerebrospinal fluid, spinal studies. It depends on the context of use, us being careful to abide by that, and having a honest data and conversations about what exactly they're going to do with that information. And only then, I think we should be using this biomarker or this test, as well as any other test for that matter.
Thank you, Nilüfer, for this very thoughtful answer. You also pioneered the use of amyloid-related endophenotype to uncover genetic pathways. But endophenotype means different depending on whether you are a clinician, a neuroscientist, or policymaker. Could you help us clear up ambiguity and then sketch how you see the field transitioning from this pathology anchor endophenotype to omic-derived one that might better capture Alzheimer heterogeneity and potentially also enable more accurate diagnosis even in the early stages of the disease when patients are asymptomatic.
Sure. So let's start with defining the terminology endophenotype. So actually, this is a term that goes back to 1960s. It was a term that was first introduced in the context of Drosophila genetics. And the goal was to contrast the obvious and external phenotypes, the exophenotypes, to those that are microscopic and internal, the endophenotypes. Then in the 1970s, the term endophenotype was applied in psychiatric genetics, specifically first in schizophrenia genetics, and it was to describe molecular outcomes of gene activity which lead to disease. And the term started to be used in a more widespread fashion in the 21st century in other complex conditions, including neurodegenerative diseases. So what we mean by endophenotype is several things. So first of all, it's a measurable outcome. Second, there is an implicit understanding that these endophenotypes have a genetic component, that at least to some extent, they are driven by a genetic component, and either the endophenotype or the genetic component also drives some aspect of disease. So think of amyloid beta, for example. There's a genetic component to amyloid beta. You can measure amyloid beta as an endophenotype, and the same genetic component is also a risk factor for Alzheimer's disease. So it's not a sine qua non that the same genetic components underlie both the endophenotype and the disease, but the endophenotype and disease are somehow related to each other. So why look at endophenotypes at all? Well, the idea is that because endophenotypes are measurable, that they may lead to this precise information to be amyloid beta, tau, endophenotypes, such as gene expression levels. That's part of it. Second, when we're talking about a disease entity, let's say Alzheimer's disease, it's extremely heterogeneous. It's heterogeneous in its clinical expression, but it's also heterogeneous in how it comes to be. Today, we think that there are hundreds of genetic risk factors for Alzheimer's disease interacting with the exposome environment leading to the disease. In contrast to that, endophenotype theoretically might have a simpler genetic background, although that's an assumption, that's a generalization. And the last component of it is when you're talking about these, it's almost a binary outcome. In contrast to that, an endophenotype is quantitative, and by virtue of it being quantitative, it gives more statistical power than the genetic factors associated with it. So those were the reasons why Alzheimer's disease embraced the concept of endophenotypes. Initially, we were using, I mentioned, we pioneered the use of plasma amyloid beta as endophenotypes, but Ornit, as you point out, now we are using other more complex multiomics measures as endophenotypes. So the framework goes like this. There are myriad genetic factors that influence the endophenotype that we're measuring. And they interact with epigenetic factors. So both the alphabet of our DNA as well as the modifications of our DNA, plus interactions with environment lead to some perturbations in a measurable molecular outcome. And that measurable outcome could be the transcriptome, gene expression levels. It could be the proteome, protein expression levels. It could be metabolome, lipidome. There's other ohms, but let's stick to these ones. So then these perturbations in the transcriptome, proteome, etc., they are not occurring as a vacuum, as a one-off, but really they tell us about perturbations in biological pathways. So by studying how the whole transcriptome is changed, and by doing really deep dives, we can begin to understand what biological pathways are perturbed. Ara, you mentioned that. Dr. Swerdlow talked about mitochondrial pathway perturbation. So that's something, for example, that comes up from proteome, from metabome, from transcriptome, and there is others. Why is it important to study omics as endophenotypes? Because they are a window into the myriad changes, the myriad perturbations that occur at a molecular level in diseases like Alzheimer's disease. Alzheimer's disease isn't amyloid beta, it isn't tau, it isn't neuroinflammation, it isn't myelin deficits, etc., etc. It is, in some ways, all of these things. And by understanding what's perturbed and what are the key molecules that are important in those perturbations, we can begin to prioritize therapeutic targets as well as biomarkers. So I took you all the way from 1960s definition of endophenotypes to how we're using multiomics right now. I'm going to pause there.
No, that's quite all right. And actually, we're very much aligned on these topics, scientifically, and really sort of this whole idea really, I think, could be a whole nother discussion, possibly even a whole episode series by itself on these topics of really sort of disease complexity. But to really get back to the issues of omics derived endophenotypes as we just described, what would you say is really the minimal evidentiary package needed to elevate this sort of discovery science to sort of now something a little bit more practical for regulators, physicians, patients, sponsors that they can trust? In other words, what does it take to sort of demonstrate that analytical validity, clinical validity, and clinical utility for these kinds of biomarkers or health outcome measures?
Thanks, Ara. So you're talking about the spectrum of translational science, right? So from the standpoint of omics as biomarkers, or as tools to give us information on therapeutic targets, we are more in the T1, T2 stages of that translational spectrum. We're in the discrete aspects of it, maybe going towards a clinical packet, but most translational research, unfortunately, dies between T2 and T3. That's the valley of death, isn't it? Where discovery scientists have hundreds of therapeutic targets, possibly biomarker targets, and yet, we have very limited offerings to our patients. So your question, Ara, really is about how do we pass through the valley of death? How do we go from discovery to truly implementation science? And the FDA has clear guidelines on what would constitute a biomarker. But the question that we must ask ourselves is, those are regulatory guidelines. What can we do as investigators, big pharma, small pharma, patient advocacy groups to ensure that we pass through this valley of death and we accelerate this research? So it's going to require a number of things. First of all, we need to ensure that whatever it is that we're measuring is technically measuring the same thing. So there's a technical aspect of it. And measuring the same thing with similar parameters. So that's very important. The technical aspect of it is very important. The other aspect of it is, what does that measurement mean? So that's the context of use. Is this omics biomarker, or is this biomarker, take away the word omics, a biomarker that's diagnostic? Is that going to ensure that I'm going to diagnose somebody with Alzheimer's disease? Is that a prognostic biomarker that I'm going to be able to say, you are going to have a steeper progression? Is that going to be a theranostic biomarker, which I can also use to perhaps measure the efficacy of a drug? So I think that's the other aspect. That is going to be my context of use. And then the other thing is this easy? Let's say we pass through all of those hurdles that we identified and analyzed, an omics measure that's technically we're able to measure all across the globe. And it means the same thing in the different patient populations, and we know what it means. Have a new biomarker that tells us about a new facet of Alzheimer's disease, not amyloid, not tau, but let's say it tells us about how much you have mitochondrial damage in your brain, but we're doing it in blood. Great. So then the next step is, how are we going to implement it in the population? Is it easily accessible by the population? And does it mean the same thing everywhere? So it sounds like a lot of work, because it is, but I do think that we have a wonderful case studies in plasma biomarkers of amyloid and tau. We must remember that the beginnings of that go back to more than 2 decades ago. So you might say, well, you're doing omics research. Does it mean it's going to take another 20 years to bring them to fruition? You didn't ask me the question, but I'll answer it anyway. My answer is, I hope not. And why is that? Because we have built certain resources in those 2 decades, and they include trial-ready populations, longitudinal cohorts. We are technically much more advanced in being able to do the measurements, and analytically, we're much more advanced to be able to analyze the outcomes. So my hope is that we will see an array of different biomarkers and therapeutic targets come to bear if we work jointly together and in a principled and data-driven way.
Thank you.
Thank you.
I know Ornit has a follow-up question. I just want to punctuate something you just said, and that really is sort of the heart of what we're trying to do with this podcast series, is really to try to drive that innovation across these disparate types of data, and really sort of deliver validated tools for patients. Ornit, I'm sorry.
Well, and ending on your point regarding population, I want to just building on that and bring to diversity. Even if a signature passes dot validation study, much of our field's evidence is based on and come from Eurocentric cohorts. How do we ensure that proteomic and transcriptomic biomarkers are not just valid, but also can be generalized in terms of conceptual utility, but still stratified across diverse population? Or what specific steps in study design, data harmonization, regulatory standards do you think that are most critical in order to diversize these biomarkers?
Sure. So I think this applies to any disease, any therapeutic, and any biomarker that I think we have an obligation to be able to see whether it's applicable to broad populations. And by that, I mean different geographies, different ancestral groups, broad populations. I mean, Alzheimer's disease it is a global epidemic, isn't it? And so I think it's will be hard pressed to find any population who isn't affected by this condition, and so our solutions should be as broad as possible. Understanding that, given the heterogeneity of the disease, some solutions may be applicable to certain populations. I'm talking about there may be a group of a certain polygenic risk that entails certain biologies to be affected in some groups of patients. I'm talking about biological subtypes. And so what works in those patients as therapies may not be applicable to other patients. And I think here, Alzheimer's disease and related disorders needs to take a page out of a textbook of cancer, because the cancer field is at least a decade ahead of us. I give the same example in a lot of different contexts. We're not talking about this patient has cancer. We're not talking about this patient has brain cancer. We're not even talking about this patient has glioblastoma. We're talking about this patient has glioblastoma. That's this stage that has this epigenetic mutation that is expected to respond to X but not to Y. And that is precision medicine. For Alzheimer's and related disorders, we're not practicing precision medicine, not yet anyway. Maybe we're starting to a little bit with ApoE and the anti-amyloid treatments. We're studying conventional medicine. So for us to truly be effective in treating, in diagnosing, predicting not just onset, but progression, and predicting treatment response to Alzheimer's disease, we need to understand the omics signatures for as many, as broad groups as is possible, define the biological types, understand what omics biomarkers go with those subtypes, and design subtype-specific therapies to enhance efficacy and reduce toxicity. That's what I understand from embracing all populations to be able to treat this disease.
Thank you. And I would like to reiterate one of the points that you raised. We have a lot to learn, a lot of lessons to learn from cancer research, from the basic research through translational all the way to clinical research when we study neurodegenerative diseases, the entire spectrum of neurodegenerative diseases. So in this corner, we talk about what makes an omic endophenotype decision great. But I would like to take it to transition to our next corner. Much of that validation has been done in cohorts of large European ancestry. From your perspective, is the field really becoming more inclusive, or are we drifting towards a geographic of privilege where only a few populations are well represented? Although ClearID is an important first step and a terrific example for population diversity, how can we globalize to other countries and continents? Is there a getaway for ClearID to become international station for omic validation and their utility? And you are close to the Space Coast.
So I like the International Space Station analogy, and sometimes our consortia feels like one, although we are very grounded. we do have gravity. So I think uh, we do see a lot of evidence, a lot of growth in these international collaborations, whether that's intellectual collaborations, whether that's for validating findings. Really, it is to everyone's benefit to be working together for all of the reasons that I explained. I think there are many examples of how these global collaborations are occurring. So, for example, not an omics study, but a study on lifestyle, the finger study and the pointer study. That's an intellectual collaboration, but that's also a collaboration in the sense of the same type of study, perhaps not looking at precision biomarkers or precision therapies, but looking at lifestyle changes in affecting populations' cognition all across the world. I think that's phenomenal. That's a great example, and there are other examples in the genetics realm. For example, we have come a long way, even in the past. Past decade, I would say, not even 2 decades, we went ApoE only as a risk factor, as a very important major risk factor, to having 80 plus loci for Alzheimer's disease. And that's as a result of these international collaborations, either validating one another, utilizing their data, doing meta-analysis. And I think the studies in Alzheimer's and related disorders are quickly expanding to other populations as well. We see examples of this in, for example, Southeast countries that have not traditionally had omics type of studies. These studies are being increased in Japan, in South Korea. In Latin America also, there is significant studies, significant efforts in understanding the genetic risk for Alzheimer's disease in that ancestral background. And then in the United States also, studying many different ethno-racial backgrounds, as well as the contribution of the exposome. So all of this work is going to culminate, in my opinion, in our understanding more of this this aspects of this very heterogeneous condition, and us being able to offer therapies and biomarkers for all of us.
Yeah, that's great. Thank you, Nilüfer. And it's sort of, my mind is kind of going towards the question of infrastructure and this explosion of data, and the the pressing need to validate biomarkers across diverse populations. We're not going to need just bigger data sets, which we're accomplishing with finger, pointer, other types of these, even clear AD clearly. But we're also going to need new ways of analyzing this complexity, how to bring together the biology, the computation, and the clinical experience in ways that the field hasn't really done before. And I know your background is not computer science and any of that type of stuff, but nevertheless, looking ahead, interdisciplinary collaborations or other methodological innovations do you see as sort of the most important, or certainly the most interesting, for that next step?
Sure. So I'll give an example from Clear AD. You alluded to it. So Clear AD is a U19 that is aimed at identifying the next generation of precision biomarkers and also precision therapies for Alzheimer's disease in multi-ethnic populations. Taking advantage of a deep molecular phenotyping with multiple layers of omics, but combining that with deep phenotypes, both clinical and neuropathology phenotypes, and looking at all of this data from the lens of the bleeding edge of analytics, which includes but is not limited to systems biology approaches as well as AI approaches. And I think, if you take Clear AD, we have 13 different sites, over 100 investigators, and many different disciplines working hand in hand. And I think it's clear, no pun intended here, why we need those different disciplines when you look at the scope of what is trying to be accomplished. So this is in the context of one U19, a large U19, but if you consider the whole landscape of Alzheimer's disease, our only hope of one day eradicating this disease is if we think outside of the box and if we work across all sorts of aisles. And we have a number of advantages in the current era. One is, as I mentioned, our ability to be able to generate these data, but also the other is our growing ability to be able to analyze these data by virtue of these analytic approaches, but also by increasing ability to harness all of these data in different computational realms using cloud systems, etc. That also enables researchers to share data, of course, in a way that all the patient information is protected in a de-identified manner, and that enables researchers to ask their own questions to the data. And that amplifies the use of that data. And that, I think, is what's going to accelerate us getting from where we are, which is data, to knowledge, to viable therapies. Some things that we need to keep our eye on is how can we harness the full power of AI, and that's not just in being able to analyze the molecular data, but we can use AI to design intelligent drugs to our therapeutic targets. That's one approach. Can we use AI to design in silico clinical trials, utilizing existing electronic health records and interrogating those health records? So before we enroll any patient in a study, can we do an in silico study, perfect some of the parameters there, and then really go into our patients for clinical trials? So these are some of the variety of ways in which we can combine discovery science with translational science with clinical trials.
Well, that's terrific. And you really presupposed my next question. And so I'm going to just slightly modify it in a sense, because I think you really hit on the issues of what are some of the computational challenges that we're facing, particularly as we're thinking about Alzheimer's in a complex systems approach. One of the things that certainly as editor of Alzheimer's and dementia years ago, and now sort of working in this field between Alzheimer's, neuroscience, and computer science, is the great difficulty in trying to communicate complexity, even at a conceptual level. And so maybe you can just sort of briefly comment on your own experiences in talking with colleagues in terms of getting them to sort of, I think people acknowledge that it's a complex disease, but do they really think about it? And do they really, do they have the tools to really describe that complexity?
So I think your question gets at two important themes, in my opinion. One has to do with communicating science, right? And I think that's a key issue, because all diseases, in my opinion, but especially a disease like Alzheimer's disease, requires complete partnership between investigators, the scientists, the physicians, the healthcare system, the patients, patient advocacy groups. So that requires, I'm not going to say a common language, but at least a language where each of us can understand the gist of what we're saying. And I think, especially as investigators, I feel like we have an obligation to be able to explain our findings and where we're going next in a manner that's clear for our patients, for patient advocacy groups, for governments, all of that. And that's an area where we don't get educated on, right? I mean, we learn about, okay, so this is how synapses occur, and this is how myelin wraps around the exon, all of that, or, here's how you generate and analyze transcriptome. But how do you convey all of that to the population? And it's our obligation to be able to do that. So I'm also an educator, and this is something I'm passionate about. I serve as a PI of our KL2 program in our clinical and translational science center at Mayo Clinic. And communication needs to be taught. So there's no question about that. How important this is. What does this finding mean? And importantly, this is something we found in this population, and I'm here to explain to you what it means for all of you. We need to be able to do that as scientists, as physicians. So that's part of it, on communicating complexity. The other part of it is communicating with one another, and that requires a very open-minded perspective. I think we have already moved on from siloed ways of doing science. I'm not going to say nobody does it in a siloed way, but I think for diseases like Alzheimer's disease, we have moved on from siloed ways of doing science to this collaborative, transdisciplinary scenario. So let's not move back to the siloed era.
Yes. Great.
Yes, yeah. And I think you already touched our next question, that we need to take further step and basically increase the interdisciplinary collaboration across different fields. And as we're talking about bridging together biologists, data scientists, clinician, and also, as you mentioned, social scientists, ethicists, maybe also, and health system experts to the same, bring everybody to the same table so we can tackle the issue, the complexity in a way that is meaningful for the patient. But how are we bringing the patient into this complexity? What your thought there?
Yes. So I think having patient advocate representatives at the table is essential. I've seen this happen in a very meaningful way in two relatively recent platforms. One of those was the National Academies of Sciences, Education, and Sciences, Engineering, and Medicine put together a committee to basically look into what we need to do as a nation to accelerate therapies and find cures for Alzheimer's and related disorders. I was honored to be a part of that committee, and I was honored to work alongside experts in this, including patient experts. So that's one platform, and the other platform was the FDA committee for one of the anti-amyloid therapies. I served on that advisory committee where we also had representation from patient advocacy groups. In both of those situations, I learned a ton from the patient experts and patient advocacy group experts. I think we cannot make a decision that impacts 7 million people in this country, 40 million in the world, as only scientists or physicians or policymakers or all of us need to be at the table, importantly, patient experts and also patient advocacy groups. I think in Alzheimer's disease, there are strong advocacy groups, which is great, Alzheimer's Association being one of them. But we need to insist for the presence of patient experts at the table.
For sure.
Thank you. And that basically brings us to another key aspect of complexity. Most current models are focused on risk and pathology, but complexity science highlight the importance of protective pathways. So namely what we call resilience and reserve. And we are being very purposeful in blending the two terms of resilience and reserve. In Alzheimer's, these concepts are frequently co-discussed, but usually these two terms as resilience and reserve, but usually it remains broad and qualitative. From your perspective, what would it take to make resilience a measurable construct in trial? Can we define resilience endophenotypes in the same way as risk-based ones and meaningfully incorporate them into trial framework?
I think, Ornit, you're touching upon something that's extremely important, where there is some effort, but we need to enhance that effort, because you can either try to reduce a bad thing or enhance a good thing, and we need to be doing both of those things, right?
Mm-hmm. Right.
So in the current use, there are certain definitions of what constitutes resilience or reserve, or resistance is the other term. And I think very broadly, resistance is you have some kind of a risk factor, but you don't develop the pathology. And then resilience is maybe you develop the pathology, but you don't have clinical consequences despite having the pathology. So I think you could think of those that way. And then reserve is somewhat similar to resilience in that you accumulate pathology, but because of something positive that you have, something beneficial that you have, you don't show the signs of the disease, period, or you don't show it as early as you should have. So these are useful constructs, right? We have to have these constructs to be able to study aspects of disease or aspects of protection. I think that there does exist constructs that can be applied in the context of clinical trials today. We can measure to some extent the amount of pathology a person has, either using the amyloid tau biomarkers, biomarkers of neurodegeneration. But of course, I'm going to say you need biomarkers of other aspects of the disease, such as ones that Clear AD is working on. You can use those and say, okay, this group of people have all of these signatures of bad things happening in their brain, and yet clinically, whereas they should have been here, they're here from a cognitive standpoint. And that's a construct. That delta is something you can measure and you can apply. You can use it in clinical trials. You can use it in other scenarios. I think, to me, the biggest missing component of resilience, reserve, resistance study in Alzheimer's disease are the cohorts, because I think the cohorts that we have are either, these observational cohorts of at-risk folks, but except for some cohorts that are intentionally going after people who age successfully, and that includes successful cognitive aging, we don't have as many cohorts to go after. So I think that's a missing link. I think we need to have collections of biospecimens, iPSC-derived models from these resilient individuals. And I think our drug development pipelines should not only focus on bringing down the bad, but they should also focus on increasing the good. And I think by doing those things, we can take advantage of both the yin and the yang of these diseases, including Alzheimer's disease.
And it's the whole field is a multidimensional complex. So the complexity has even another dimension, and time is the key. As we know, Alzheimer progress over time. And in the clinical trial, we basically look at multiple time points. So how might longitudinal study, and bringing another dimension here, help us to understand how resilience or reserve or resistance act or change over time, and potentially as a factor of aging here in this group of diseases?
We have, I mean, longitudinal studies are key. These observational studies with deep clinical phenotypes, deep molecular phenotypes, and patients who give their time, the participants who give their time, and then make the ultimate donation gift of their brains. That's priceless, right? These are the groups that enable us to understand this condition. I think in addition to these cohorts that are actively being studied, we need to, coming back to Ara's point, be able to harness this messy data called electronic health records, right? They're not like the observational longitudinal studies that have their very nice parameters and in-depth information, but electronic health records can also have a very powerful resource of information, and they are longitudinal in many ways. They're not, they don't have this nice parameter around them, but by virtue of numbers, we may be able to glean very important information from them. So I think and then the third thing I'm going to say is, again, human-derived models. We need to develop better and better models of aging, both successful aging in a dish, as well as risk in a dish. So I think we do have the technologies, and we can harness all of these aspects together to understand both the risk aspect and the resilience, reserve, resistance aspect of disease.
That's great. And if we could measure, or maybe rather than say measure, parameterize resilience, reserve, and resistance, how would you see it being used as a prognostic tool to stratify patients, as a predictive factor for treatment response, or even as a trial endpoint in its own right? And I'm going to ask a really unfair question, which is, what's the pathway to move from this resilience, reserve, resistance from theory into something that patients, clinicians, and sponsors would actually understand and responsibly and ethically use?
So I think, to some extent, we are using this not in a clinically actionable manner, but as an observation, right? I'm going to give an example from internal medicine, family medicine. I'm sure anyone who's gone through medical school has been given examples of somebody's aunt who smoked a pack a day, drank and wasn't educated in all of this, and all these risks, and they died at age 90 without a heart attack, stroke, etc. So there is something about that. So the concept has been around for a very, very long time. But what you're talking about, Ara, is can we measure it? Can we, to use your term, parameterize it and apply it in the clinical setting? So this is theoretically possible. How is that possible? To the extent that we can measure the presence of risk factors, and there's the today, there's the future. So let's be futuristic. In the future, let's say in the next 5 to 10 years, we're going to have a blood test. From the blood test, we're going to be able to say, okay, this is the polygenic risk score. And then, we've done the proteome, transcriptome, methylome, metabolome, lipidome analysis, maybe not of everything, but of the top things that came off of these omics-based studies, let's say, including Clear AD. We measured those, and based on those, here is your risk score, okay? Based on that risk score, this is where we would expect your cognition to be, let's say, but this is where you're at. Or you may say, based on this risk score, we expect you to have this much amyloid, this much tau, this much neuroinflammation, this much neurodegeneration. But you don't have those things. Instead, you're much better than that. And that delta becomes your reserve, that's your resilience. That's one way of applying it. Or you could say, you have those risk factors. We're going to start you on a drug cocktail to address all of those perturbed pathways. And then you see the patient back. And because of all of these longitudinal studies, you have an expectation of how much you expect them to decline, but they declined much less than that. So then that delta becomes the gain that you have achieved with the drug cocktail that you gave the patient. So this is how I envision that the fluid biomarkers, the omics biomarkers, as well as the deep phenotypes are going to be parameterized for us to say whether there is a useful drug outcome or not useful drug outcome. But it requires not just omics, not just other biomarkers, but also deep phenotypes and longitudinal follow-up.
Thank you. And to bring this vision and how we envision a thing gonna be in the future into fruition, if you had to pick two levers to make proteomics and transcriptomics real and accessible for clinical trial design, would it be infrastructure, data harmonization, new policy models, or anything else that you think or envision?
I think right now, the biggest hurdle at this stage is we're good at, as I said, T1, T2, but the clinical application is the hardest hurdle. And I think that's because of time and cost and implementation difficulties. The way for forward thinking about this is I'm not going to be able to do multi-omics on millions of people, right? So I need the top things, the most informative things, to be able to say to somebody, you have this much risk of a heart attack. We're not doing a whole lipidome study. We're doing LDL, HDL, cholesterol, triglycerides, right? So we need the LDL, HDL, total cholesterol, triglycerides version of the most informative markers, okay? And then we need to develop technologies to be able to assess them rapidly and broadly and cheaply. So I think the first order of business, so if I had two levers, one would be prioritize, validate, validate. Go from the ohm to your top hits, the most informative things. That's lever number one. Lever number two is technology. Having found those top things that are going to give you information on the perturbed pathways and all of the different types of biomarkers that we discussed at the beginning of this podcast, then the technology to be able to measure them cheaply, easily, everywhere, and fast. Those are the two levers that I would pick.
Okay.
Thank
Well, that's fair. So let's flip it around a little bit and to the patient or to the frontline clinician asking and they've stumbled onto this on YouTube and they're wondering what difference will these tools make for me? What would you say to them?
Yes. So research seems slow because it is slow. And I see patients, and this is a question my patients ask me all the time. You do research, is it going to impact me? How is it going to impact me? So this is what I will say. I think sometimes looking back helps us for perspective. If we look at the last 10 years of Alzheimer's disease, maybe that gives us information about the next 5 years of Alzheimer's disease. Past 10 years gave us numerous hundreds more targets. Last 10 years gave us new FDA-approved anti-amyloid treatments. They're not the one all or be all. I see them as a start, not the end, but as a start of other therapies to come. Last 10 years gave us new fluid biomarkers that we're currently using in the clinic to distinguish patients with Alzheimer's pathology, and that we're using for guiding patients towards these anti-amyloid treatments. So last 10 years gives us hope. It gives me hope for the next 5 years. So the next 5 years is going to be about what other aspects of this disease am I going to target? What are my next clinical trials going to be? What are my next biomarkers beyond amyloid tau neurodegeneration going to be? And that's going to impact us because it gets us closer to treating different facets of this disease, being able to access as many patients as we can, and being able to gain information on the different facets of this disease. So I'm very hopeful, but that hope is based on data.
Nilüfer, thank you so much for joining us. Your work has shown how genomics, endophenotypes, and clinical insights can come together to sharpen our approach to complex diseases. This has been an incredibly rich discussion.
Indeed, it was a pleasure to host you here, Nilüfer, in our first episode. And for our listener, stay tuned for episode 5, where we will continue this journey by going under the hood of data pipeline and computational approaches. Our guest will be Dr. Michael Lotz, a professor of neurology and pathology at Duke University, who specialized in computational biology approaches to unravel the genetic basis of Alzheimer and other neurodegenerative diseases. Mike brings both an academic and industry and pharmaceutical perspectives, having led computational genetics effort at GlaxoSmithKline before he joined Duke University.
Exactly. Mike's interdisciplinary expertise makes him the perfect next guest as we ask, how do we actually build computational data frameworks that will allow proteomics and transcriptomics to move from basic science discovery into scalable clinical trial tools? Until then, I'm Khachaturian with Ornit Chiba-Falek. Thank you for joining us, and we look forward to traveling these roads with you again soon.